Sharks are vital to ocean health, but as long-lived predators, they're especially vulnerable to environmental contaminants. At Clemson University's Reproductive and Ecotoxicology Lab, we're investigating how pollutants like mercury and PFAS accumulate in sharks, not just across species, but also across life stages.
Hello, I'm Mel Walker, a master's student in the Repro-Eco Tox Lab. Our research looks at how feeding behavior, environment, and age interact to influence contaminant exposure in sharks and what that means for their health and survival. With the help of the South Carolina Department of Natural Resources, we're studying fine-tooth and sandbar sharks, two species with very different diets. The fine-tooth shark is a dietary specialist, feeding mostly on menhaden fish, while the sandbar shark is a generalist that eats a wide variety of prey. We use ethical catch and release methods to collect shark body measurements, a blood sample, and a small muscle biopsy. We sample individuals across age classes, from neonates to juveniles to adults, to understand how contaminant levels change as sharks grow, mature, and shift their feeding behavior. Back in the lab, we test the blood and tissue samples for mercury and PFAS, chemicals that accumulate over time and can interfere with growth, reproduction, and immune function. Because these sharks live for decades, it's important to understand how early life exposures compare to those in older individuals. Additionally, we are analyzing 19 known local prey species to see how diet contributes to contaminant exposure. This helps us evaluate whether age-related changes in feeding habits lead to a higher or lower pollutant accumulation. Understanding how contaminants affect sharks at different life stages gives us insight into their long-term health and the health of the ecosystems they're part of. The overarching goal is determining how environmental challenges shape their future at every stage of life.
Our team is also expanding this work through a project funded by the Save Our Seas Foundation. Alongside Dr. Megan Holst at UC Davis, Dr. Taylor Chappell, and the Big Fish Lab at Oregon State University, we're studying two other shark species, the broad-nosed sevengill and taupe sharks, across different age classes in two different estuarian systems. In this study, we're comparing contaminant levels in sharks from San Francisco Bay, a heavily urbanized estuary, and Willapa Bay, a comparatively pristine coastal ecosystem. By sampling individuals of different sizes and life stages, we're building a more complete picture of how age, ecology, and environment intersect to shape contaminant risk. This research helps identify vulnerable life stages, informs conservation and management strategies, and contributes to broader understanding of how pollution impacts long-lived marine predators. By studying contaminants across shark life stages, we're not just learning about exposure, we're uncovering the lifelong effects of pollution on one of the ocean's most essential predators.